So now we are going to talk about the gastrulation, the mammalian gastrulation. Okay, so mammals are related evolutionary. Okay, so evolutionary, if we talk about the mammals are related to birds, right? We say from reptiles, we have both birds and mammals. They came out, right? That's what, am I right, William? Yeah. I think you are more evolution side. So that's how they happen, right? So if it happened like that, that means that somehow there should be some hint of the similar kind of activity in the development, right? So when we look at the mammalian gastrulation, so mammalian gastrulation is actually very similar to birds, but the bird has a big yolk, right? We all eat egg and we see big yolk there. In human, there is no yolk as such, right? So then it means there is no yolk in mammals. So therefore, when you are developing, you need food, right? So therefore, there should be modifications in the embryo to get nutrients. Should be modifications in the embryo to get nutrients. And we say that nutrition, so nutrition from where it is coming directly from the mother. Therefore, an organ should be developed. So if you want to continue with the development, you need to have source of nutrition. If you want source of nutrition, there is no yolk. You need to get connection with the mother, right? So that means you need to develop an organ for that. And that organ is coming from trophoblast. So what trophoblast does, trophoblast develops through various stages. So there are multiple stages there. Develops through various stages to become chorion, And we say, what is chorion? So chorion is embryonic derived portion of placenta. Okay. Now, so trophoblast also induces placenta, the uterine cells, or uterine tissue, to form the maternal portion of placenta, and that is called decidua. Decidua. So the placenta is essentially two parts. One is coming from the embryo, that is a trophoblast make them, that is called chorion. And the other part of placenta is coming from the uterine wall or uterine tissue, and that is formed because that is induced by the chorion. Chorion tell it to make, and that part is called deciduum. Okay, so placenta have two parts, chorion and deciduum. Now, how the chorion formation takes place? Formation of chorion. Now, so what is happening there? Let me draw this thing again. So we are saying we have these cells right there, and we have these ICM cells right there. These are your trophoblast, and these are your ICM. Okay. Now, the ICM cells, what they are doing, they are developing embryo. Now things are happening side by side, right? So and these trophoblast cells, these cells are developing chorion. Okay. Now, so these things are happening side by side. So, but I cannot do this thing and this thing side by side. So we are going to talk separately each thing, right? So initially we are going to talk about how the chorion is formed, and then we'll talk about how the ICM is developing the embryo. So what is happening there, that if this embryo need to develop, this embryo should survive, right? And this embryo can survive only if this trophoblast make chorion, and then chorion tell placenta to make the uterine cell to make decidua, so a placenta can be formed, right? So this is critical right there. So how this thing happens? So these trophoblast cells, where they are, so let's talk about this. Well, let me draw right here. Right here, you have right here, this is your endometrium. And we are saying right here are cadherins, and from here you have these integrated. Okay. Now, what is happening, we did talk that this trophoblast cell, they start secreting right here is the enzyme, the proteases. Okay. So what happened? So they are secreting proteases. So trophoblast secrete proteases. So trophoblasts are secreting proteases here. So what they are doing actually when they are doing that, let's go to the next step. So they are kind of secreting the proteases, and they are kind of pushing them like this. So these are the trophoblast cells are coming there. Okay. So so this is again your endometrium. end. So these cells are dividing when they're going like this. So these cells right here are dividing. The trophoblast cells are dividing. They are secreting proteases and dividing. And they're pushing down. You see right here they were on the surface. They're kind of starting burning it down, right? So at this stage, these cells, which are trophoblast cells, they are no more called trophoblast cells. We call these trophoblast cells now when they're trying to bury themselves. These are called cyto. So these are now making cells, and these are called cytotrophoblast cells. Now what happens? These cells now, what they do, they start dividing more rapidly, and they divide in a way that you have nuclear division but no cytokinesis. So, what happened? These cells divide rapidly. So what they have nuclear division, but no cytokinesis. What does mean? It means that what is going to happen, that you are going to have a situation where you are going to have so this embryo is actually going further down. So you had some cytotrophoblast, and now what you have here is you have actually very big cells like this. These big cells are there. Now remember, I said that nuclear division is taking place but no cytokinesis. It means that the cells, nucleus is dividing but cell is not dividing, right? So that means you will have the cells here with multiple nuclei. Cells with multiple nuclei there. Okay? And these cells which are with multiple nuclei, these are called syncytotrophoblast. Okay, so, and these syncytotrophoblast, what they are doing, they are making further progression of embryo in uterine tissue by secreting more proteases. Okay, so far it is okay. So the cells, they start dividing, but they don't divide in a way that the cell does not divide, nucleus divide. Syncytoblast are formed, and they are further burning down because they are secreting more and more of the enzymes, proteases, digesting it. Now what happens? So we are going further. So now what happens that, so this is your so they have kind of putting them down further, and your cytotrophoblasts are there. And then you have more of syn cytotrophoblasts there. Okay, now, so while they are doing this, two things are happening. When these cytotrophoblast cell, they started burying it. When they're burying it, while that time, because now this is a tissue there, right? This tissue should have blood vessels, right? So when this is burying there, what they are doing, these cytotrophoblast cells, they are modifying uterine blood vascular system. So what they do, that uterine blood bathes embryo, right? It's blood everywhere, the embryo. Now what happens here? So that system is going on, it's burying, okay? At that point, when this reaches further, these syn cytotrophoblast they secrete, not syn cyto, sorry, this cytotrophoblast. Cytotrophoblast, they secrete paracrine factors. So they had modified the blood vessels here, right? Now at this stage, they secrete the paracrine factor. And what those paracrine factors do, they attract blood vessels. So what happened? From here, the maternal blood vessels are being attracted there because they are secreting. So these cells are secreting paracrine factor. And those paracrine factor, now this is everything is the placenta, right? So those paracrine factor are attracting these blood vessels here. So blood vessels are coming here. Blood vessels are coming here. Blood vessels are coming here. Now, these blood vessels are coming from the mother. Now what happened? So if you think that this is a, let me draw the cross section. Think about this is the cross section of the blood vessel. So these cells which are, these are called endothelium cells. Endothelium cells, they line the blood vessels. Now these endothelium cells in these blood vessels should be from the mother, right? But what happened? These cytotrophoblasts, they start replacing the endothelium cells with themselves. So instead of now this endothelium cell from the mother, what is going to happen? They will be replaced by the cytotrophoblast cell. So cytotrophoblast cell, they will start lining the blood vessel which have now entered inside. Making sense so far? And then so remember, when this structure is being formed, same time the gastrulation here has started, okay? And when the gastrulation starts, you know the mesoderm form, right? So some of the mesoderm from here will move outside the embryo. And that will be called extra embryonic mesoderm, okay? So then extra embryonic mesoderm joins the cytotrophoblast. So, what happened that the blood vessel system is formed by the mesoderm actually. So initially it was cytotrophoblast, then mesoderm also joined it. So when mesoderm is joining it, that means that blood vessel system is formed. Making sense? So what is happening that, so cytotrophoblast join to form the blood vessels. Now what you have? Now you have the blood vessels. What you're having, what is the purpose? To carry nutrients from the mother. Okay. Now, here this is developing, and what is the placenta part? Placenta part was all the, this is, here is the placental tissue, right? And those blood vessels from placenta are coming here. Making sense? Not the placental uterus. So this whole portion containing the blood vessel from the mother, from the embryo, now they are joined, right? Those plus the chorion plus this tissue of the mother placenta. This portion will be called decidua. This will be called chorion, and this total will be called placenta. So what is happening? Let me go back so that everything is clear. We are saying that this embryo that was coming, that was ready to implant, has integrin on it and they have cadherin on it. And we are saying these trophoblast cells, they start secreting proteases. When they start secreting proteases, they are digesting extracellular matrix and they are trying to take itself in. And they can take itself in by dividing these cells more. These cytotrophoblast cells start dividing, and they actually started digging inside. At that time, we call these cells as cytotrophoblast cells. We don't call them trophoblast cells anymore because they are to the next stage. And then what happened? These cells are secreting proteases, all those things are coming down. And these cells also modify the blood vessel of the placenta so that they are kind of bathing this whole coming in embryo. Now what is happening? These cells, cytotrophoblast cells, some of these cells start dividing rapidly. And they divide in a way that no nuclear division, or multiple nuclear division, but no cytokinesis. So that cause these cells, new cells are formed. These are called cytotrophoblast cells. Cytotrophoblast cell further produces more proteases and basically bury itself inside the tissue. When this thing has happened, these cytotrophoblast cells, they start secreting the paracrine factor. And those paracrine factors, they attract the blood vessels from the uterine tissue. And when those blood vessels are now moving from here to here, at that time, the tissue or the endothelial cell which were lining the blood vessel here, they are totally replaced by the cytotrophoblast. And the mesoderm, extraembryonic mesoderm come out, and that mesoderm also line these vessels and form the blood vessels. Now these blood vessels are completely formed and they are joined to the uterine tissue, and the uterine part of the tissue which is contributing these blood vessels to provide nutrient to the embryo, that is called decidua. And then this part which is formed, this is called chorion. And this whole thing is called placenta. Now think about that. A placenta is an organ that is formed by the contribution of the tissue from the two individuals who are genetically different. This is important thing to remember. Like, this is the organ which is formed from the contribution of the two genetically different organisms. Mother is genetically different from the embryo. They are not same genetically, right? So this organ is formed. Now, what chorion is doing? Chorion, which was here, formed this vesicle, kind of the fuses with decidua, so you have a placenta formation. Now, what is the purpose, function of placenta? So if you talk about the function of placenta, number one, nutrition. So placenta is providing nutrition, you're getting nutrition, right? Number two, it produces hormone, and that hormone is progesterone. And what is the purpose of progesterone? Progesterone tells uterus to retain the pregnancy. That was the initial thing, to retain the pregnancy. So progesterone that retain pregnancy. And the third thing is, it blocks mother's immune system from rejection of the foreign substance. And foreign substance here is the embryo that is developing. That's not the mother's body, right? So there can be immune rejection. So that's what placenta is doing. So, any question here? Okay, so talk to each other for a minute, then we'll go further to make sure that everything is clear.
Like the sagittal valve replace the outer cells, and then the extra endogenous. Is it endogenous? I think that was the placenta. Is it going to attach from the embryo and the decidual comes from the mom? Any questions?
Any doubts here? You're okay, Mason?
Yeah.
You're okay with it? You guys have any doubts here? Okay, good. So now, we know how that placenta is now formed, right? So now we are going inside and we are going to talk about, again, we are talking about gastrulation. I'm just giving them separate names, right? So this is development of ICM. So when the things are happening outside, few things are happening inside also, right? So what happened that So these are the cells. Now, you know what is happening to the trophoblast, right? So we are not worried, I'm just drawing it like this, you know all those changes are happening there, right? So what happened, we have these cells right here are the so these are the ICM cells. Okay. So the first thing happened here, the first segregation of ICM cells. Okay, this is interesting. So what happened that ICM forms two layers of cells. So ICM formed two layers of cells. Now, I'm not drawing everything the tropho, I'm just trying to show you what is happening inside, okay? So what is happening, it formed two layers of cell. One layer, I'm just drawing four cells, there might be more than that. And then there is another layer of the cells. So remember, what is this? This is blastocell. Okay. So the cell, it formed two layers. The cells which are in contact with blastocell and the cell which are away from the blastocell. The cells which are in contact with blastocell, these cells are now called hypoblast cells. Okay. These cells are going to give primitive endoderm and going to form yolk sac. And these cells do not contribute to embryonic development. But these cells, the cell do not contribute to embryonic development, but they help regulate the migration of epiblast cells during gastrulation. Okay, so what is happening there? We are saying that cells have divided into two layers. Cells which were in contact with blastocell, they become hypoblast cell. They're going to give primitive endoderm. And these cells, they do not contribute to embryonic development. I mean, their cells do not contribute. I mean, they're not going to form any organ system or anything like that. But what they're going to do, they're going to help the migration of epiblast cells during the gastrulation process. Okay, so because they form the yolk sac. Okay, so now, so that means what we are having a hypoblast formation taking place. These cells which are there, the red cells, these cells are called epiblast cells. Okay, so you can say epiblast layer because this is one layer, this is hypoblast layer, two layers of the cells. Now, epiblast cells, they are going to give the embryo. They are going to give the embryo. Okay. Now, so these cells are not contributing anything in the embryo formation now. Then why some cell become, so we said, remember everything is changing. When it was blastocyst, we were saying, hey, every cell of ICM, inner cell mass, can give you a new organism. We said they are pluripotent. Here, we are changing that statement because this is no more a blastocyst. They have moved from one stage to the next stage. So that means these out of those ICM cells, these cells, the hypoblast cells, they are no more pluripotent, right? They can only give the primitive endoderm. Making sense? So things are changing as the development is taking place. Now, then whether to be epiblast or hypoblast, how that is determined. What cells are going to be hypoblast, or what is going to become the epiblast. Now that depends on the timing. That depends on the timing. And timing is that when the cells become the part of ICM. At what time they become the part of ICM? Okay? So cells, those become ICM at 8 to 16 cell division. Okay? Those become pluripotent epiblast cells. And the cells become ICM at 16 to 32 cell stage. They become hypoblast. Now remember, when they were ICM, they still had all the characteristics. They were all pluripotent. But somehow, later distinction is based on at what time they entered in that group. Okay? So now, the cells which are going to become epiblast cells, they express OCT4 and nano. Okay? And the cell which are going to become hypoblast, now they are no more stem cells. If there are no more stem cells, then they should not express the transcription factor of the stem cells, right? So they express GATA6, and they will become primitive endoderm. Okay, now this is important thing. Remember, when we're talking about germline specification, we said those cells are set aside in the early development outside the embryo proper. And we're showing allantois membrane there, we're showing chorion, and we said they are in the extra embryonic endoderm right there. Right? So from here, right from the epiblast cells, some cell will be thrown out, and we'll talk about those structure later, in somewhere in this group of cells, just saved outside, right? And the cells which are going to become cells, going to become germ cell, will retain OCT4. But the cells which are not going to be Germ cell, they will lose the OCT4 later. So remember that germ cell have the capability to give you a new organism. And in your experiments in the lab, you are doing one of those germ cells, right? Those are testicular carcinoma cells. So those are the germ cells. Now those germ cells are pluripotent because they have OCT4 in them. Making sense? So each step, you are losing that potency when you're going from one step to other step. So that's why, again, I'm saying the same thing. Having, say, embryonic stem cell, these are now no more embryonic stem cell because they cannot give you everything. You have these hypoblast cells coming out there. Making sense there? So that's how and this kind of like potency decreases as the development proceeds. So we are saying that ICM cells, they essentially segregate in two layers. The layer closer to blastocele become hypoblast cell. Hypoblast cell is going to give you primitive endoderm that is going to form yolk sac. And these cells are not going to contribute anything to the embryonic development, but these are going to help the migration of epiblast cell during the gastrulation process. So the gastrulation process has not started yet, okay? And then we are saying that why to become epiblast or hypoblast, that depend upon at what time you entered into the ICM. If you entered into the ICM at 8 to 16 cell division, that first division from when this, before the compaction, right? Compaction is 8 cell, then compaction start. Right there, if you have entered in the center, then you have become the epiblast cell. If you are in the next division, 16 to 30 cell, you will become the hypoblast. So kind of fate is written right there. And then we are saying that hypoblast cell, they are going to express GATA6. No more OCT4, no more NANO, no more SOX2. Whereas the epiblast are expressing OCT4 and NANO. And then we are saying the cell which are going to become germline cell, they will keep on OCT4 throughout. Whereas the epiblast cell, they will have NANO, and of course SOX2 will be there, and the SOX2. Making sense? Any question? Okay, so you can talk to each other for a second, and we'll go further.
I'm going to get the primitive endoderm. Going to form the yolk sac to contribute to embryonic development, but they have regular migration of epiblast cells during gastrulation. So whether it becomes an epiblast cell or the hypoblast cell depends on what becomes part of ICM. The ones that become part of ICM, 8 to 16 cells become epiblast, when 16 to 32 cell stage become hypoblast. Epiblast cells express OCT4 and NANO, and the cells will become germ cells which contain OCT4. And then hypoblast cells express GATA6 and become permanent endoderm.
Any doubt here? Any question? You guys are okay? Okay now. So we are going to talk about formation of amniotic cavity. Now, we all know amnion is there, amniotic cavity is there, that is a fluid-filled cavity, and that is going to protect the developing embryo from any shock, right? So far gastrulation has not started. So you can see all the preparations are going on for the development of the embryo. So those preparations were not there in the amphibian because they are in the water, right? They are not worried about. Those preparations are not going to happen even in the birds and reptiles, but those preparations are happening in the mammals. Because this is developing inside, you need food, there's no yolk, we had the development of coil, right? And then we want to protect it from the shocks and all those things. So what happened? Still no gastrulation. So this is the stage we did talk about. We said this is the embryo right there. And we said, hey, we had these type of cells there. We had these cells were there, which were your epiblast cell, right? And then we said, hey, we had these cells. So you had this epiblast cells, and then you had these hypoblast cells, right? Now, these epiblast and hypoblast cells are there. So what happened that these epiblast cell, so remember when we were talking about the gastrulation, we said there are multiple movements in gastrulation take place. One movement we said what delamination, we said splitting. We said one layer form and we feel like two layers are forming, they split from each other. And that's what happened here, still not gastrulation, but these cells, they divide and they split in two layers, okay? So epiblast cells, they split in two layers. Okay, they split in two layers. So what is going to happen? You will have like this. These are your hypoblast cells right there, and your epiblast cells, they're right there. They split in two layers. Okay. So epiblast cells split. And then what happened? These cells, which are here, so we have like one layer of cell here. So these cells which are here, they actually start producing the same thing that would happen in the cavitation process. So they have the sodium ion. So what they do, they start putting sodium ion right here. Okay? So right here what you have is the sodium ions start coming there. And when the sodium come there, so first sodium ion, then followed by water. Okay, when that happens, what you get is, you get a situation like this.
27 hours at attention.
And right here is your okay. So this cavity is derived. Now this is it. Fluid filled cavity called amniotic cavity. So fluid filled cavity is formed. Alright? So what is this? This is a shock absorber. For developing embryo. So far it is okay? Okay. Now, these cells which are here, they are derived from epiblast, but they are not epiblast cells, right? And they are the part of the amniotic cavity now. So that means, therefore, if we have to say that amniotic cavity is derived from epiblast. These cells are derived, right? So these cells are forming lining of the epiblast. Now, what we have now? Now we have, this is hypoblast. Hypoblast, and this is epiblast. We have two layers of cells, right? And their cells, and this is called bilaminar germinal disc. Bilaminar germinal disc. So far it is okay? Now this is the time for hypoblast cells to do something. So then the hypoblast cells, they say, okay, why should we stay there like this? They say we will also divide. So these hypoblast cells, now the hypoblast cells, hypoblast cells start dividing and line the blastocyst. So these cells were there, they start dividing. And they start dividing and they essentially start forming another layer here. Okay. Now, this has gone one further step. Now hypoblasts are no more hypoblast cells. Okay. So at that time, these cells which are right here, which are attached to the epiblast cells, these cells are called visceral endoderm. So these cells are called visceral endoderm. And the cells which are lining above the blastocoel, these are called parietal endoderm. Okay. And these together are called exocoelomic membrane. And this whole structure, which is right here, this structure is called yolk sac. So this structure, this thing is called yolk sac. So two sacs are formed there, amniotic sac and the yolk sac. Making sense? Now the stage is set for gastrulation. So now, these remaining cells, which are just this many cells, these are still the epiblast cells. They are still the epiblast cells. They are going to embryo. They are going to give you embryo. So now what did we talk? We said that you had the two layers of cells in the beginning, the hypoblast cells and epiblast cells. Then the epiblast cell, they decide to split in two layers, right? They split and form two layers. And then these cells start secreting sodium ion, which is followed by the water, and this whole area create a cavity called amniotic cavity. There's a fluid-filled cavity derived from epiblast cells. But these lining, which are from the epiblast cells, they are no more. pluripotent. They have become the part of the amniotic cavity. And amniotic cavity acts as a shock absorber for the embryo. So that's what this is happening before the development of embryo starts. And then we are saying that these hypoblast cells, they also start dividing and they start migrating along the ceiling of the blastocoel and essentially line this whole thing. Then these are no more hypoblast cells. We are saying the cells next to the epiblast cell are called visceral endoderm cell, and cells which are lining the blastocoel ceiling, those are called parietal endoderm cell, and these both are called exocoelomic membrane. Okay, and this cavity which is formed right there, this is called now yolk sac. So we are saying two different type of sacs are formed: amniotic sac and this is yolk sac. Now this two things, the hypoblast and epiblast together, these will be still called bilaminar germ disc. Okay, and now the stage is set for the gastrulation, and gastrulation will occur with the help of yolk sac in these epiblast cells. That is going to give you. Now these epiblast cells are going to give you all. Now think about this is very different than the circular amphibian embryo, right? Where things were outside, they are going in, there was endoderm down, and these things were up. Now you still don't have endoderm, although you have parietal and visceral endoderm, but these are not the part of the embryo. This is kind of like saving somewhere your germ cells. So making sense, and those germ cells we know will migrate later inside that. Making sense? Any doubt here? Yes.
So once the epiblast cells split into two layers, that's when they lose their pluripotency?
Epiblasts still have pluripotent, but these cells are not pluripotent, right? Because pluripotency means you need to give every cell of the body. So they still can give you every cell type of the body. That's what they're going to give. They're still the cell, but they have not made anything of the body yet. Right? Making sense? Okay. Any question? Yes.
So the parietal endoderm is just the top lining one?
Yes.
And then the visceral
Is next to epiblast.
just the middle?
Yeah, next to epiblast. Any doubt? Okay, do we have time to start the castration or we're done?
It's 10:03.
Oh, sorry, okay. So we will meet tomorrow in the lab and you will see, actually I have observed the immunochemistry. It has worked, but it has not worked. It has worked to take the pictures, there is no doubt about that. But it has not worked the way it used to work. The reason is not your fault. We have our freezer, kind of, there was a thaw, and it was kind of like thaw for the antibody